Hello, this is a recording for Biology 122 Anatomy and Physiology lecture. The chapter we're going to be covering today is the reproductive system, the male reproductive system. And I thought I silenced my notifications, but apparently they're not working. So I will share my screen now. We're going to talk about the reproductive system today. And this video, I will only cover the male reproductive system. A lot of this in the male reproductive system, at least for my class, we've already covered many of these structures. So this diagram right here gives a sagittal view through the male pelvis. If you recall from your chapter where we covered the urinary tract, this is the urinary bladder and the urethra. The first part of the urethra that goes through the prostate is known as the prostatic urethra. Then the part of the urethra that goes through the perineal muscles is known as the intermediate or the membranous urethra. And then the urethra that goes through the penis is known as the spongy or the penile urethra. All right. As part of the male reproductive system, the sperms are made in the testes, and testes are held in a sac known as the scrotum. The testes are intra-abdominal at birth, and they descend in a full-term fetus. Sometimes they may descend soon after birth. If one or both the testes do not descend after birth, they are known as hidden testes or cryptorchidism. And clinically, sometimes doctors will wait for the testes to descend. And in case they do not descend in a timely manner, they are surgically made to descend, because testes that remain inside the body tend to be at a higher temperature, which is detrimental to sperm maturation and formation. One of the reasons why the testes are held in the scrotal sac is because the temperature in the testes needs to be a little lower than the core body temperature. All right, so the testes are held in the scrotum, and we'll go into the detailed structure of the testes in a little bit. But sperm produced in the testes, it goes through the epididymis and then into the spermatic cord. That ascends up the scrotum, goes through the inguinal canal into the abdominal cavity, goes behind the ureters, and widens, the spermatic cord widens to become the ampulla of ductus or vasa deferens. And this gland here is known as the semivesical gland. The duct from the semivesical gland and the vasa deferens ducts join together to form the ejaculatory duct. The ejaculatory duct opens into the prostatic urethra. So the testes and the sperms, along with the seminal fluid secreted by the seminal vesicles, enters into the urethra. So only in the male, the reproductive system and the urinary system merge together, if you will, in the prostate. And the sperms are ejaculated through the urethra. The other structures associated with the male reproductive system is the prostate gland, the bulbourethral or the Cooper's glands. And the other primary structure associated with the male reproductive organ is the penis. We have the root of the penis here, the corpus spongiosum, and the corpus cavernosum. The corpus cavernosum, as the word suggests, cavernous means cave-like, It is a very vascular structure and can hold large quantities of blood under certain circumstances. And towards the external urethral orifice, the penis has a glans penis, which is, and the end of that is known as the corona or as the crown, and the skin that covers the glans penis is known as the prepuce or the foreskin. In certain cultures and religions, the foreskin is dissected or is cut. And there are different reasons or beliefs for why that is done. In cultures that do not cut the foreskin, those individuals should be trained as they grow up to be able to retract the loose foreskin and to clean under the foreskin, because secretion known as smegma can collect under that. And smegma is a precancerous condition and can cause carcinoma of the penis. So that is one sort of application, if you will, of this part of the male reproductive system. So just to make sure I've covered all the structures here, we did talk about the scrotal sac, the testes, the epididymis, the prepuce of the foreskin. The spermatic cord is also known as the ductus or vasa deferens. So yeah, we've covered all of these structures. Moving on to the next slide. So this previous slide was a model of the male reproductive system. This is what the male reproductive system looks like in a sagittal section of a real human body. So as you can see, the structures may not be as easy to identify as they were in the previous model. So this here is the urinary bladder, and right below that is the prostate, and this is the prostatic urethra. These here is the seminal vesicle, and if you will recall, when we did the dissection of rat, when we were doing the digestive system, and we did the dissection of rats, we did look at all the other systems. And I did ask you to keep an eye out for other systems like the reproductive system. And we had seen these structures. They were the seminal vesicles in the rats. They were pretty large. So here's the ductus or the vasa deferens. And this is where the vasa deferens joins with the duct coming out of the seminal vesicle and becoming the ejaculatory duct, which enters, opens into the prostatic urethra. Oops. All right, this is the intermediate or the membranous urethra going through the perineal muscles, and then the membranous urethra, the spongy urethra going through the body of the penis. The scrotum is a sac of loose skin underlying the subcutaneous tissue that contains the testes. Internally, it is separated into two compartments. So every individual has two testes, and they are separated by a compartment. And the separation is done by a scrotal septum, and the muscle is known as the dartos muscle. Let me zoom this in. All right, so this here shows the scrotal sac, and the little indentation made on the skin because of the septum in between is known as the raphe. Here is the scrotal septum, and the testes, as you will notice, do have several layers around them. The closest layer is the tunica albuginea of the testes. And they have, again, like many other layers as we've seen in other organs, they have a parietal layer and a visceral layer. So you will notice that the tunica vaginalis has a parietal and a visceral layer, and we will see that more in detail in some of the figures coming up next. The muscle that holds that is seen in the scrotum is also known as the cremaster muscle. And the cremaster muscle can help the testes ascend and descend a little bit depending on temperature and certain other environmental factors. The spermatic cord is where the ductus or the vasa deferens ascends from the testes, goes through the inguinal canal. The inguinal canal has a spermatic cord, also has the nerves passing through it, the arteries, and the veins. And the veins are more in the form of a plexus, known as the pampiniform plexus of the testicular veins. The artery that supplies blood to the testes is the testicular artery. The testes have an autonomous nervous supply, so they have an autonomic nerve. Right here, you will see this is the internal oblique muscle and the epineurosis of the internal and the external oblique muscles coming right up here till the fundibulum ligament of the penis, which is the root of the penis. And this here shows the cross section through the penis. It has two corpora cavernosa of the penis, these two structures seen on the two sides, on the dorsal surface. And on the ventral surface is the spongy penis. The corpus spongiosum has the spongy penile urethra passing through it. And we will get into more details of the structure of the penis a little bit later. The testes are paired. They're oval glands in the scrotum, partially covered by the tunica vaginalis. One of the clinical applications of paired testes is sometimes they can get twisted. It is one of the emergency conditions clinically. It's extremely painful, excruciating pain, and it is an emergency condition in which the twist needs to be fixed, or blood supply to the testes can be compromised and can be detrimental to the health of the testes, and they will need to be removed if they undergo gangrenous damage. So testes, they are paired oval glands in the scrotum, partially covered by the tunica vaginalis. Internal to the tunica vaginalis is the connective tissue capsule. The tunica albuginea extends inwards to form the septa that creates the two compartments, the seminiferous tubules. And we'll get to the details of what these seminiferous tubules are, a more detailed structure of what's inside the testes, the structure of the testes. They are divided into lobules, and in each lobule are the seminiferous tubules, and that's where the sperms are made. So the seminiferous tubules, they carry the sperms that are produced within them, known as the spermatogenesis is the process that produces the sperms, and then they're carried out of the testes. So looking at this diagram here, which is a sagittal section through a testes, and I'll zoom in quick here. So in this sagittal section here, you see how there are little lobes formed in the testes. These lobes are formed by the tunica albuginea and make little septums that separate these lobes. And within the lobes, these twisted structures, these are the seminiferous tubules. And sperms are made within the seminiferous tubules. And we will see a magnified image of the seminiferous tubules in a little bit and see how sperms are made. But this is where sperms are made, and they're carried through these straight tubules out of the seminiferous tubules through this structure known as the retitestes. And from the retitestes, they go up into the epididymis. The epididymis has two parts. This part superiorly is known as the head of the epididymis, and here inferiorly is known as the tail of the epididymis. The sperms go through the epididymis and up into the vasa deferens. And vasa deferens goes through the spermatic cord into the inguinal canal and then into the abdomen. All right. These ducts coming out of the retitestes going into the epididymis are known as the efferent duct. And so that's the head of the epididymis. This is the body of the epididymis here. Here's the tail of the epididymis. These are the septum, lobes, the tunica albuginea, and we did talk about the tunica vaginalis. Right here is the tunica vaginalis. It has two layers. This one here is the visceral layer, and the outer layer is known as the parietal layer. So the parietal layer of the tunica vaginalis goes all the way in, turns around, and comes back, and this is the visceral layer. So remember, the visceral and the parietal layer are basically the same structure that turns around. Very similar to the pleura in the lungs, the pericardium in this heart, and the peritoneum in the abdomen. All right, and along with the vasodifferens in the spermatic cord, we also have the nerve supply, the autonomic nerves, testicular artery, and the venous plexus. Again, a clinical application of the venous plexus is sometimes these veins can get engorged if the patient has what are they called? Engorged veins. Varicose veins. If the patient has varicose veins, these veins in the testes can get engorged and varicose too. So that is another clinical condition that needs to be managed. All right, so that previous slide was a model of a testes. This here shows you what a real testes in a dissected human body looks like. So this is a testes where just the tunica albuginea has been removed. That's the testes. Here's the epididymis. This is the head of the epididymis, the body of the epididymis, and the tail of the epididymis. And from here, the ductus deferens goes into the spermatic cord. This is a sagittal section through the testes, showing the seminiferous tubules. Let me see if I can zoom in. There you go. So this is the seminiferous tubules, and you can see the structure of the lobules is really not very distinctly seen in this diagram here, but that's where your lobules will be, and the seminiferous tubules. This right here is the rete testes, and then you have the epididymis up here, the head of the epididymis, and then the tail, and then going up the spermatic cord going into the vasoductus going into the spermatic cord. Now looking at a cross-section through the seminiferous tubules. So these right here that you saw, these tubules, if you were to take a magnification of a cross-section through one of these tubules right here, that's what you're seeing in here. This is a cross-section through the tubule. All right, and you see how there are cells over here that are attached to the outer layer, and then these cells on the inside, these are the spermatids. So the cells that are attached to the outer basement membrane, these are your spermatogenic cells. These are stem cells that will differentiate to become spermatids, which will mature and become the sperms. All right, and here's a model of the same structure. Right here in the outer layer, just above the basement membrane, in the tissue, you have the interstitial cells. These cells are the ones that produce testosterone. You have the blood vessel, the basement membrane down here, and attached to the basement membrane are the spermatogonium. Now, the spermatogonium is diploid. Diploid means 46 chromosomes. Diploid cells have 46 chromosomes. All right. Now, the spermatogonium undergoes mitotic division and becomes the primary spermatocyte. So one spermatogonium will divide, mitotic division, and become two primary spermatocytes. One of these primary spermatocytes will continue to stay attached to the basement membrane, while the other one will undergo a mitotic division. The first one was mitotic division, and this one is a meiotic division, meiosis, where from diploid it becomes a haploid cell. So instead of having 46 chromosomes, the secondary spermatocyte only has 23 chromosomes. So it undergoes meiosis 1 and meiosis 2, and one primary spermatocyte cell with 46 chromosome results in four spermatids that are haploid, that have only 23 chromosomes. These spermatids will undergo transformation and become a sperm cell or a spermatozoon, which has a head, a neck, and a tail. And we will see a detailed structure of a sperm in just a minute. So we talked about the interstitial cells. These are the cells that produce testosterone. We have the basement membrane. We have the spermatogonium here, and these are the sustenticular cells. The sustenticular cells are the ones that have receptors that attach to receptors for luteinizing hormone, follicle stimulating hormone, and testosterone that stimulate or regulate the production of sperms. All right, and as these sperms mature, they are released into the lumen of the seminiferous tubule. So again, going back to this diagram, the spermatogonium are attached to the basement membrane around the periphery of the seminiferous tubules. And as they undergo initially mitotic division and then meiotic division, they become sperm cells or spermatozoons. And as they cross through the seminiferous tubules, making their way through the retitestes and into the epididymis, they mature and become mature sperms. All right, we'll watch a short video on spermatogenesis that will explain the meiotic and mitotic division.
Spermatogenesis is the production of male gametes, or sperm cells. The two testes are the sites of spermatogenesis, or sperm production. Within the testes, the specific sites of spermatogenesis are the walls of the seminiferous tubules. Spermatogenesis begins with the spermatogonia cells at the onset of puberty. Following DNA replication during interphase, spermatogonia are diploid stem cells having 46 sister chromatids, or 92 DNA molecules. Stem cells are less differentiated cells that can undergo mitotic cell division, thus producing cells that undergo further differentiation. One daughter cell stays near the basement membrane and remains a stem cell. The other daughter cell loses contact with the basement membrane. Upon losing contact with the basement membrane, the second daughter cell is squeezed through the tight junctions formed by the Sertoli cells. This daughter cell undergoes biochemical changes and differentiates into a primary spermatocyte. Each primary spermatocyte has the same number of chromosomes as a spermatogonia cell. Each primary spermatocyte enlarges, and the first meiotic cell division begins. As a result of the first meiotic division, two haploid secondary spermatocytes are formed. Each secondary spermatocyte has 23 chromosomes. Since each chromosome has two sister chromatids, each cell contains 46 DNA molecules. All of the secondary spermatocytes undergo the second meiotic division. As a result of the second meiotic division, 4 haploid spermatids are formed. Each haploid spermatid has 23 chromosomes. However, since each chromosome consists of only one chromatid, one copy of DNA, then each spermatid contains 23 DNA molecules. The final stage of spermatogenesis is spermiogenesis. Spermiogenesis is the maturation of haploid spermatids into sperm, also termed spermatozoa. There are a number of changes that happen to spermatids during spermiogenesis. The spherical spermatids turn into longer, slender sperm. An acrosome develops on the side of the nucleus, which condenses and elongates. A flagellum also develops, and mitochondria multiply. Finally, the sperm cells are released from their connections to Sertoli cells and enter the lumen of the seminiferous tubule. Sertoli cells secrete fluids that nutritionally support sperm, as well as drive them along the seminiferous tubule toward the epididymis.
So this is just showing a quick overview of the mitotic division. And then meiosis 1, where they go from being diploid cells to being haploid cells, and then meiosis 2, where the 2 daughter cells then become 4 daughter cells, all of which are haploid. And then the spermatogenesis, where the daughter cell transforms to become a spermatozoa. All right. So the primary spermatocyte undergoes meiosis 1 to become 2 secondary spermatocytes, which are haploids. Meiosis 2 takes place, and the secondary spermatocyte becomes 4 spermatids. All right. And this is, again, just an overview of the same process that we saw earlier, where the spermatogonia becomes the primary spermatocyte at the end of mitosis, and then you have your meiosis 1, resulting in 2 secondary spermatocytes, which are haploid, and then meiosis 2, you get 4 daughter spermatids, and these spermatids undergo spermiogenesis to become spermatozoa, again, all of which are haploid, and the physical transformation is the condensation and formation of the head, the neck, and the flagellum, or the tail. And these then are separated and released into the lumen of the seminiferous tubules. All right, the sperms are designed to reach and penetrate the secondary oocyte in order to achieve fertilization and create a zygote. The head contains a nucleus, which has 23 chromosomes. The acrosome covers the head, and it also contains enzymes that help with the penetration process once the sperm reaches the secondary oocyte. The neck contains the centrioles that form the microtubules that make up the rest of the tail. And the neck also contains the mitochondria that make ATP, and we've learned how mitochondria make ATP, which give the sperm its locomotion, the energy to move. The principal piece, the tail, the flagellum, has a principal piece and an end piece that make up the tail. So if you look at the image of a sperm, this is an enlarged image of a single sperm. So here's the head, which has the acrosome, the nucleus, which contains the chromosomes. Here's the neck. This middle piece contains the mitochondria that produce the ATP. And then the middle piece and the tail. The middle piece and the principal piece and end piece together make up the tail. And as you can see, the difference in the principal piece and the end piece. The end piece is much thinner, if you will, and doesn't have this outer layer. And the flagellum is sort of like a propeller. It propels the sperm forward. Hormonal control of testicular function at puberty, the gonadotropin-releasing hormone stimulates the cells in the anterior pituitary gland. So the gonadotropin-releasing hormone is produced by the hypothalamus. It stimulates the anterior pituitary gland to produce 2 hormones, the luteinizing hormone and the follicle-stimulating hormone. The luteinizing hormone stimulates the cells in the testes to produce the testosterone. It's the follicle-stimulating hormone that stimulates spermatogenesis and formation of the sperms. All right, let me zoom in. So at puberty, the hypothalamus secretes the gonadotropin-releasing hormone, which releases the follicle-stimulating hormone and the luteinizing hormone. The luteinizing hormone stimulates secretion of testosterone by the interstitial cells. And remember, we saw the interstitial cells when we looked at the detailed structure of the testes, the Leydig cells that secrete the testosterone. Testosterone and follicle-stimulating hormone both together stimulate spermatogenesis in the testes. The sustentacular cells, which we saw again when we looked at the detailed structure of the seminiferous tubules, the sustentacular cells, they secrete androgen-binding proteins, ABP. All right. Once we have the required blood levels of testosterone and follicle-stimulating hormone, both of these have a negative feedback loop, both to the anterior pituitary and to the hypothalamus, to regulate the amount of hormones in the blood. All right, so there's a negative feedback loop to maintain homeostasis. So as you can see here, the luteinizing hormone that acts as your ligand, you have the receptors on the interstitial cells that bind and stimulate the release of testosterone. Testosterone undergoes metabolism and can become dihydrotestosterone, or DHT, which is responsible for male pattern development before birth, and enlargement of the male sex organs, expression of the secondary male secondary sexual characteristics at puberty, like facial hair growth, the regressing hairline, the hair on the ears, the deepening of the voice. All of these secondary sexual characteristics occur after puberty. And even anabolism, or growth, or protein synthesis. If you recall from anatomy ANP1, we talked about how when we look at bones, how do we differentiate between male and female? And we did look at a lot of the features in the bones are more prominent in male bones than in the female bones, and that is all because of the role of testosterone. This is all part of the anabolic action of testosterone on the body. So testosterone and dihydrotestosterone, they produce several different effects. They're involved in the prenatal development of the male fetus, development of the male sexual characteristics after puberty, development of sexual function, also stimulation of anabolism. A negative feedback system controls the blood levels of testosterone, again, to maintain homeostasis. All right, and this is again a quick overview of how homeostasis is maintained and blood levels of testosterone and other hormones are maintained. Is when you initially have a stimulus or any kind of a disruption in the homeostasis, in this particular case by increasing blood level of testosterone. So when there is high blood levels of testosterone, the receptors, the cells in the hypothalamus that secrete gonadotrophin hormone. So when you have high levels of testosterone, they will decrease gonadotrophin releasing hormone in the portal blood. This will have its effect on the anterior pituitary gland, which will decrease the secretion of luteinizing hormone into the systemic blood, which will again have less effect on the interstitial cells in the testes, so they will secrete less testosterone. So remember, we started off with an increase in blood testosterone level. This causes the response of that stimulus is a decrease in blood testosterone level. This again has a return to homeostasis when the response brings blood level of testosterone back to normal. All right, so this again has a negative feedback loop. So that's how blood testosterone levels are maintained. So there's a system of ducts in the male reproductive system. Sperm and fluid travels from the seminiferous tubules to the straight tubules and then to a network of ducts called as the rete testes. The efferent ducts carry the sperm to the epididymis. The sperms mature in the epididymis, and sperms have a short life. So they can start undergoing degenerative changes within the epididymis if they are not ejaculated. These degenerated sperms are reabsorbed. The epididymis propels the sperms into the ductus or the vasa deferens. All right, this is a posterior view. So this is as if you're looking from behind. This is a posterior view of the accessory organs of the male reproductive system. Again, remember, you're looking at it from behind. So this is the hip bone. So these two hip bones are forming the pubic symphysis in front of the urinary bladder. You're looking at the posterior surface of the urinary bladder. So the spermatic cord, or the the vas deferens comes up through the inguinal canal into the abdomen, goes in front of the ureters. This dilated part of the vas deferens is known as the ampulla of ductus deferens, or ampulla of vas deferens. And here, these are the seminiferous vesicles. The duct from the seminiferous vesicle joins the vas deferens and becomes the ejaculatory duct. The ejaculatory duct opens into the prostatic urethra. So the part of the urethra that's going through the prostate, right here, this is the prostate gland. This whole thing is the prostate gland. So the part of the urethra that's going through the prostate, that's where the ejaculatory duct opens into the urethra. And this is where the male reproductive system and the male urinary system share the structure, the urethra. Then you have the intermediate or the membranous urethra going through the perineal muscles or the urogenital diaphragm. These right here are the bulbourethral or the Cooper's gland, and we'll get to their function in a bit. They too have a little duct that opens into the urethra. Right here, this is the base of the penis. So we have the crux penis and the bulb of the penis. The crux of the penis goes down to become the corpora cavernosa of the penis, and the bulb of the penis becomes the corpus spongiosum of the penis. The ductus or the vas deferens, it exits the tail of the epididymis. It ascends through the spermatic cord into the pelvis. It goes through the inguinal canal. And I think I've talked about this in my previous lectures. The inguinal canal in males, because of the spermatic cord going through it, makes men more vulnerable to inguinal hernia. So men who have predisposing conditions like obesity, chronic cough, or are involved in occupations that require them to do heavy weightlifting, heavy labor, anything that causes constant and persistent increased abdominal pressure makes them vulnerable for inguinal hernias. So the spermatic cord goes through the inguinal hernia into the pelvis. Then it loops over the ureter, passes over the side and down the posterior surface of the urinary bladder, and then joins the duct coming out of the seminiferous of the seminal vesicles and opens into the prostatic urethra. So this is again the posterior view of a male accessory organ in a dissected body. The spermatic cord ascends up the scrotum, contains the ductus deferens, testicular arteries, veins draining the testes, the autonomic nerve system, the lymphatic vessels, and the cremaster muscle. The spermatic cord and the ilioinguinal nerve pass through the inguinal canal, which originates at the deep inguinal ring and ends in the superficial inguinal ring. So these are the two ends of the inguinal canal, the deep inguinal ring and the superficial inguinal ring, where the spermatic cord enters and exits the inguinal canal. All right, and this is pretty much the same diagram as we saw initially, the very first diagram that we saw in the beginning. I don't think there's anything new here, so I'm just going to skip over this. The ejaculatory ducts arise from the junction of the duct from the seminal vesicle and the ampulla of the ductus deferens. The urethra is the duct that shared by the reproductive and the urinary system. So both semen and urine are passed through the urethra in the male only. The urethra passes through the prostate gland, where it's known as the prostatic urethra, the deep muscles of the perineum, known as the intermediate or the membranous urethra, and the penis, also known as the spongy urethra. All right, taking a slightly closer look at the structure of the penis here. All right, so starting up here, this is the urinary bladder, the internal urethral orifice. This is the prostatic urethra, and the orifice of the ejaculatory duct that opens the ejaculatory duct, which contains the contents of the spermatic cord and of the seminal vesicles. They open and empty into the prostatic urethra here. Then you have your intermediate or your membranous urethra. You have the Cooper's glands here. The Cooper's glands will secrete secretions into the urethra during sexual arousal and during ejaculation. And we will get to the contents of all of these glands in a little bit towards the end here. This is the bulb of the penis right here, which becomes the corpus spongiosum. And right here is the crux of the penis. Both the bulb of the penis and the crux of the penis form the root of the penis, which attaches it to the body. The corpora cavernosa, as you can see, cavernous means cave-like. So it's a very vascular tissue. It has a capacity to hold large amounts of blood when vasodilatation occurs. And right here is the spongy or the penile urethra, the external urethral orifice. The corpus spongiosum forms the glans penis, and the corona here, the skin that covers the glans penis, is known as the prepuce or the foreskin. And the procedure to dissect the foreskin is also known as circumcision, which I talked about earlier. In certain cultures and religions, they do remove this foreskin. And in cultures that don't, those men should be educated and trained to retract the foreskin and to clean under the foreskin to prevent collection of sphagnum under that, because that is a precancerous condition. All right, now talking about the different glands and their secretions. The seminal vesicle gland, their secretion is alkaline and viscous fluid, which contains fructose. Fructose is a source of nutrition for the sperm, because the sperm, once ejaculated, can stay alive in the female body for up to 7 days. So for those 7 days, those mitochondria and the cells need nourishment. So seminal vesicle fluid secretion provide the fructose required for that. Prostaglandins and clotting proteins are required. The prostate is a single donor-shaped gland that secretes a milky, slightly acidic fluid containing citric acid, proteolytic enzymes, acid phosphate, and seminal plasmin. Oops, sorry. The bulbourethral or the Cooper's gland, they secrete an alkaline fluid during sexual arousal that neutralizes the acids from the urine, and also secretes mucus for lubrication during sexual activity. So this is, again, only during sexual activity, the secretion. An average person's semen is a mixture of sperm and seminal fluid. An average ejaculate contains anywhere from 2.5 to 5 milliliters of sperms. And in that 2.5 to 5 milliliters, there can be anywhere from 50 to 150 million sperms per ml. That's the average range. People who may have below that range are said to have oligospermia, and oligospermia is a clinical condition very commonly responsible for primary infertility. So couples who are trying to conceive and have a baby and are not able to have a child, many a times, one of the first few procedures done is to do a sperm count on a male's ejaculate. Very often it is found that the sperm count is low. And a sperm count needs to be within this normal range for successful fertilization of the oocyte. So oligospermia is a common cause for primary infertility. The pH of the ejaculate is about 7.2 to 7.7. The penis contains urethra and is a passageway for semen and for urine. The semen, as we've seen earlier, is composed of 3 cylindrical masses. It has 2 corpora cavernosa and 1 corpora spongiosum. The glans penis is the head of the penis that is covered by the prepuce or the foreskin. All right, so here, this is the dissection of transverse section through the penis. We'll look at the model first here. So this is the transverse section through the penis. This is the dorsal side. That's the ventral side. So on the dorsal side, you will notice these two here, This is the corpora cavernosa of the penis and has deep artery of the penis. And the corpora spongiosum is on the ventral side and contains the spongy urethra. The tunica albuginea for just the corpora spongiosum is separate from the tunica albuginea going around the corpora cavernosa. And up here on the dorsal side, you will notice, and here's a magnified view of that, we have the deep dorsal vein and the superficial subcutaneous dorsal vein. So you have two veins here, a superficial vein and a deep vein. There is a dorsal artery. There's also a deep artery found within the corpora cavernosa. Now, during sexual arousal, this is the structure that undergoes significant amount of vasodilatation and collection of blood and enlargement, which is responsible for penile erection. Moving on to what a dissected model in a human looks like. So that is the skin around the penis, that is the dorsal surface, this is the ventral surface, this is the corpora cavernosa, the deep artery. You see how tiny it looks over here, the deep artery, and this is why it's known as corpora cavernous. It's got cave-like large caverns, if you will, which gives it its name, the corpora cavernosum. And this is the corpus spongiosum with the spongy urethra going through, and this is towards the ventral surface. And here you have the deep dorsal vein, and the superficial dorsal vein is way up here. All right, an erection is brought about by parasympathetic innervation, leading to vasodilatation of the arterioles in erectile tissue. So large amounts of blood enter the tissue into the dilated blood sinuses. Ejaculation occurs due to the powerful release of semen due to sympathetic stimulation. The bulbospongiosus, the ischio-cavernous, and the superficial transverse perineal muscles. These muscles contract to force the semen out from the urethra during ejaculation. Neuronal control, or the circuits involved in erection. The stimuli for that could be erotic thoughts, erotic sights, erotic smells, erotic sounds, or mechanical stimulation of the penis through the mechanoreceptors can send stimuli to the brain and or to the spinal level. Once the hypothalamus and the limbic system is stimulated, it sends descending inputs from the brain to the parasympathetic preganglionic neurons that send, again, autonomic stimulus to the erectile tissue in the penis that causes penile arterioles to dilate and the penile veins to compress, which leads to the erection. Watch a quick video on hormonal control of the male reproductive function.
Hormonal mechanisms that influence male reproductive function involve endocrine tissues contained in the hypothalamus of the brain, the anterior pituitary, and the testes. In the hypothalamus, certain neurosecretory cells secrete gonadotropin-releasing hormone. Gonadotropin-releasing hormone is carried by the blood to the anterior pituitary, where it binds to receptors on gonadotrophs and stimulates the cells to increase secretion of two gonadotropic hormones, luteinizing hormone, or LH, and follicle-stimulating hormone, or FSH. LH and FSH are transported in the blood to the testes. LH and FSH both indirectly affect spermatogenesis. LH binds to receptors on Leydig cells between seminiferous tubules and stimulates the secretion of testosterone. Under the influence of FSH and testosterone, Sertoli cells produce androgen-binding protein. ABP binds to testosterone and maintains high levels of the hormone near the spermatogenic cells. Under the influence of testosterone, Sertoli cells nurture the stem cells and the developing sperm cells. Testosterone stimulates the final stages of spermatogenesis. In addition, testosterone is converted into dihydrotestosterone, another hormone that may promote sperm cell formation. Testosterone and dihydrotestosterone both bind to the same intranuclear receptors. Together, the androgens regulate male prenatal development and the development of male sexual characteristics. Sertoli cells release the protein hormone inhibin when the level of spermatogenesis required for male reproductive functions has been attained. Inhibin is delivered by the blood to the gonadotrophic cells within the anterior pituitary. Inhibin acts on the gonadotrophs to reduce FSH secretion and thereby decreases the rate of spermatogenesis. Similarly, testosterone acts in a negative feedback manner on anterior pituitary gonadotrophs to suppress the secretion of LH. The negative feedback effect of testosterone also involves its effect on the hypothalamus, the result of which is decreased secretion of gonadotropin-releasing hormone. Decreased secretion of gonadotropin-releasing hormone causes a corresponding decrease in the secretion of both LH and FSH. The decrease in LH causes a decrease in the secretion of testosterone by the Leydig cells in the testes. Low levels of testosterone reduce spermatogenesis. Conversely, an excessive decrease in testosterone allows the hypothalamus to increase its secretion of gonadotropin-releasing hormone. This secretion causes an increase in anterior pituitary secretion of LH and FSH, and an increase in secretion of testosterone by the testes.
So here I've added a slide, which is not there on the slides that were posted on D2L, and this is just an extra tidbit for everybody who actually listens to the lecture till the end. So some clinical applications with the male reproductive system. Erectile dysfunction is a very common disorder seen, especially after the age of 45. Men between the age of 45 to 50, about 45 to 50% of men between the age of 45 and 50 can have erectile dysfunction. There are two different types of erectile dysfunctions. So for everyone who has listened to the lecture so far, here is an assignment for you for extra credit. Send me an email with two different types of erectile dysfunctions. Give me those two and you'll get 2 points in extra credit. Oligospermia, I already talked about that, when the sperm count is low and is a common cause for primary infertility. Benign prostatic hypertrophy, we've talked about it in the past when we talked about urinary system. It is a benign enlargement of the prostate seen in middle-aged men. And what it does is, because the prostate enlarges, it pushes upon the urethra, on the prostatic urethra. And it can cause frequency, urgency, and it can cause a slow stream of urine, and therefore it takes them longer to complete the act of maturation, and they may have large post-maturation retention volume of urine, which sort of makes the cycle of frequency even worse because they're not even emptying the bladder every time they do maturate. Again, this is another very common disorder in men. Prostate cancer, we've talked about that, and we have androgen scans to check the levels of prostatic hormones in the blood to detect prostatic cancer in an early stage. And one of a relatively more rare condition is carcinoma of the penis. And as I said, there are many predisposing factors for carcinoma of the penis, one of them being collection of smegma because of not maintaining good personal hygiene and retracting the prepuce and cleaning behind the prepuce. All right, so with that, we end our male reproductive system. My next lecture will be on the female reproductive system. All right, so I will end here. Thank you very much. And send me your email with the two kinds of erectile dysfunction to get extra credit. Take care. Trying to stop this.